The presented protocol is used for high-throughput screening and identification of inhibitory and non-inhibitory nanobodies targeting electrogenic transporters reconstituted in protoliposomes or present in membrane vesicles. Designing transport assays can be difficult in multiple cases, especially if labeled substrates are not available. SSM electrophysiology allows studying the effect of nanobodies on transport for virtually any electrogenic transporter, since SSM electrophysiology doesn't require labeled substrates.
Nanobodies have been investigated for their usage in medical applications. This technique can help screen potential inhibitors targeting specific electrogenic transporters of humans or human pathogens.
To begin, take 10 clean tubes and transfer 10 milliliters of the non-activating solid supported membrane, or SSM buffer, into each tube. To prepare the activating SSM buffers, add the substrate into the tubes using a series of concentrations around the expected half-maximum concentration. Start the SSM software and let the machine initialize automatically. Set the saving path for data, confirm by hitting the OK button, select the standard Initial Cleaning Protocol in the workflow options, and click Run. Next, mount the proteoliposome-coated chip on the socket. Move the arm to lock the chip, and close the mounted chip with the cap. Select the program Capcom in the workflow and let it run to determine the conductivity and capacitance. Confirm that the conductivity is below 5 nanosiemens and the capacitance is between 15 and 35 nanofarad.